Today's lecture, chapter 46, starts with an overview of parasitology. This chapter has numerous tables that we're not going to cover in detail, and while we're in this chapter, we'll refer back to some of these as we talk about the organisms. But there are tables with descriptions of common groups of parasites. Table 46.1. Table 46.2 has body sites and parasite recovery. Table 46.3 has specimens, body sites, collection, processing. That goes on for several pages, 46.3. Then we also have tables 46.4 with epidemiology. That goes on for several pages. 46.5 has clinical findings and versus a normal host versus a compromised, because like with anything else, people that are compromised, immunocompromised, whatever word you want to use, are more prone to having different, more severe symptoms or more serious symptoms. But that's in 46.5. 46.6 talks about pathogenesis. 46.7 recommendations for stool testing. 46.8 of the stool collections, 46.9 fecal fixatives. And then we've also got several figures starting on page 621 that have the eggs of our helminths. We're going to go over those more when we get to the helminths. 46.10 has diagnostic specimens and positive findings. That's several pages. There's also pictures of some of our other specimens, not specimens, other organisms. There's a chart on 46.11 about smears and staining. And again, we're going to get into more of those details when we get to the actual organisms. 46.12 has antigen detection testing. So we're going to refer back to these. There's a section 46.13 on our arthropods and how we look at those. This link right here is the link to the CDC website. If you Google CDC and parasites, there is just a regular link that's a little harder to get to the alphabetical directory. So this link with the DPDX in it, if you want to Google it and say CDC parasites, just find the link with DPDX. That's the best one to see the images and the life cycles of the parasites. It's going to be a good reference for us as we go through this. Okay, so overview. Parasitism exists. It's a relationship that's going to benefit the parasite itself, the parasitic party, but it's going to be harmful to the host. Just because it's harmful to the host doesn't mean it always causes disease. Sometimes it does, sometimes it doesn't, but it still causes problems for the host, even though it's not a disease, because it's taking nutrients. They can live on the host, in the host, and the host is going to be another living organism. They can live outside the host, things like the arthropods, lice, ticks, mice. The protozoa and helminths, the parasites we're going to be studying, live inside the host. Remember our terms facultative, obligate. Obligate parasites have to be with a host to survive. Facultative means they can be away from a host, but they can be away from the host and live independently, but when they're on a host, they are considered parasitic and they do feed from that host and take nutrients. In medical parasitology, we're going to study three categories. The protozoa, that's what we're studying in this unit. The next unit will be our helminths. So our protozoa is the only one that's really considered a true microorganism. We can only see it microscopically. Our helminths and arthropods we can see macroscopically. Our helminths are worms, but we rarely diagnose them by the part that we see with the unaided eye macroscopically. We're going to diagnose these helminths with their eggs for the most part, and those eggs have to be seen microscopically, not macroscopically. Arthropods. We usually don't study the arthropods Um, as far as the diseases they cause, we're going to study some arthropods because they are sometimes the means of transmission of the parasite to us as host. Also, sometimes just part of the life cycle, even if they do not transmit it to us, part of the life cycle of some parasites has to go through another host besides us as humans in order to multiply and reproduce and then eventually make us sick or make us infected. So we're going to look at those. Um, most of how in the clinical micro lab that we diagnose parasitic infections is by observing the life cycles. We're going to see different life cycles of those. Um, very hard to diagnose and see because they are very small. Um, so when we look at the life cycles, I already mentioned there may be more than one host. That CDC website has a life cycle for every parasite we're going to study, and it will show you if there are other hosts involved in that cycle. The reservoir host is usually the source of infection. The definitive host is where the adult stage lives. That's usually the sexual stage or the sexual stage of the cycle. An intermediate host, this is when the larval or asexual stage lives in an intermediate host. So that is part of that cycle. An accidental host, what is that? This is when the organism is living inside of a host that's not the usual host. A lot of times, if a host, either us or another animal, us as humans or another animal, has this parasite um, in it, feeding off of that host, this may not be the normal host that it's usually in. So it may be a dead end for the parasite. It may not can continue its life cycle. So at that point, it can't continue to the environment or into another host and keep reproducing. It dies off at that point. We talked about this in bacteriology. We never want to confuse cause and transmission. A disease is caused by a type of parasite. Back in microbiology, we talked about diseases are caused by the bacteria. Transmitted is the way that we get sick from the disease or the way we get it. We ingest it, we're bitten by something, it's injected into our body, various ways that the parasite can be transmitted to us so that we become sick. So we have our protozoa, we're about to study those, our amoeba, our flagellates, our ciliates, our sporozoans, our coccidia, and our microsporidia. Next we'll study our helminths. With our helminths we have two classifications, nematoda and platyhelminths. The nematoda are roundworms, the platyhelminths are flatworms. The platyhelminths are further subdivided, we'll see that in the next unit, into cestoda and trematoda, or cestodes and trematodes. Pentastomodes and acanthosophilia, we won't study much about those, just a little bit. And then the arthropoda we'll study, as in not necessarily how to identify those, but how those serve as transmission, as the way diseases. Okay, so when we start to look at our protozoa, we have a few that are multicellular, but most are unicellular organisms. We generally classify our protozoa by how they move. And when we say how they move, that even includes no motility. So we look at, are they modal? What mode do they use for that movement or locomotion, or do they lack motility at all? So there are pseudopodia, false feet. Some have flagella, some have cilia. We're not going to study the protozoa that do not infect us as humans. There is a group of protozoa that are not considered parasites. They're what we call pond protozoa. When we look at our protozoa that infect us as humans, most are obligate, so they cannot live outside of a host. but a few are facultative. They live outside of a host, but if they enter us as humans, they can cause infection or disease. Most of our protozoal infections are acquired by ingesting the organism or inhaling the cyst or the spores. Our helminths, which will be the next unit after this, are what we call worms. We sometimes see the worms or pieces of the worms. That's the adult stage, or maybe even the larval portion in the specimen. But usually we're going to diagnose these by seeing the egg in the specimen. Helminths are multicellular. This is our group I mentioned earlier that are not really considered microorganisms because we can see the worm, but we do observe those eggs microscopically and see that life cycle. We have the roundworms and the flatworms, which are the nematodes, and the oh my goodness, the cestodes. Sorry, I messed that up. The nematodes are the roundworms. The platyhelminths are the flatworms, and those are divided into cestodes and trematodes. Usually we get these by ingesting the larval form of the organism that contains the egg. We can either be infected by the larvae entering our skin by penetrating it, or an infected insect, some sort of arthropod bites us and the larvae enters us. We'll learn later how when the larvae enters us, how that life cycle moves through us. So arthropods, again, we're only going to study some insects, some arachnids, some crustaceans, because they're usually either the cause of our disease, they can be the intermediate host. Some of these are the definitive host of the organisms, and we end up possibly being an accidental host. So we'll look at some of those. Vector. A vector is when the organism, the arthropod, picks up the parasite and deposits it somewhere on our body. It can either be mechanical. I'm sorry, I said that wrong. The vector can give us the organism either by being a mechanical vector, biological vector, or in some cases can be both. If it's mechanical, the vector picks up the parasite on its legs, its wings, somewhere on its body, and deposits it on a surface or even on food. We come along and we eat the food, or we touch the surface, get it on our hands, then put our hands in our mouth when we eat, or near our eyes, and then it gets into our body. Occasionally with the mechanical, it can deposit it on our skin somewhere else, like on our face, and we rub our face then, rub that feces, or rub that just where it's deposited it from landing on us into our eyes. Biological is when the pathogen multiplies or matures in the body of that arthropod, and then it transmits it to us that way. Again, we're rarely going to ID arthropods. There might be a rare occasion where we are asked to look at something under a stereoscope or under a microscope, but we don't really identify it as such as part of the diagnosis. Okay, so when we start to look at transmission, we talked about skin penetration, we talked about ingestion. There's also venereal transmission. Trichomonas vaginalis, one of our protozoa, is sexually transmitted. Then, of course, ingested, we have things like, okay, and some examples of ingesting contaminated food or water are Giardia, Cryptosporidium, and Scaris. Skin penetration. If the infective larvae penetrate our skin, such as the strongyloides or the hookworm, and bites. Some arthropods bite us. We get Plasmodium, Trypanosoma, Leishmanium. And we'll go through all of these again when we talk about the specific organisms, how they're transmitted. Diseases. Sometimes the diseases are very, very mild, or they can be very serious and life-threatening. Some just reach a status to where they're living in our body. It's just called a status quo. They're there. We don't get sick. There's no damage to our body. We may not have any symptoms. Sometimes one body site's infected. Sometimes it can be several. I mentioned earlier about the table that explains the symptoms, the disease in healthy individuals versus those that are immunocompromised, and how that affects how we respond to the infection or to the disease. Life cycles. I mentioned life cycles earlier. It's very, very important. Um, the life cycle of the parasite, how it goes through other how it goes through other individuals or other hosts, let me say it that way, and how that affects the development. Also, the parasite doesn't have a goal to cause us extreme severe illness or death, because then it has nothing to feed off of. It doesn't want to kill us, because then it loses its host and its ability to live. But sometimes that does happen. They can cause severe illness and death. Also, that life cycle, as it continues from another host to us, leaves us, goes into another host cycle. If we understand the cycle, that can sometimes help us break that cycle to control or prevent the illness, to control and prevent the cycle from continuing. It helps us with disease outcome also. With any um, study we make in the lab, any diagnosis, any testing we do, we've talked for one whole semester and part of this semester about how our specimen quality is very important. We have to have a good specimen. We have to have the right specimen, appropriate specimen. We have to process and handle it in the appropriate manner. We have to have the correct orders. We have to be trained to do this. If we do not, then we're not going to be able to identify it. It's not going to be any good. I mentioned those tables earlier, but this also mentions fixative test comments, this 46.9 and 46.10 as far as collection goes. I mentioned earlier that parasites can live during certain stages, possibly outside of a host. They can never live outside of a host, but outside of us as a human, they can live in different forms. There's adult form, larvae form, eggs, trophozoites, which is the motile form, cyst, oocyst, spores. As with any specimen really in the laboratory, the faster we process it, the better the quality of the specimen. If not, preservatives have to be used. Preservatives help maintain the characteristics when we're talking about eggs, when we get to our helmets, when we're talking about eggs. The preservatives help maintain the shape of those eggs. If not, they may deteriorate and break apart. So again, fixatives has to be appropriate, the correct fixative for the specimen and what we're looking for. You should apply the fixation, the appropriate fixative soon after the specimen is collected, and make sure it's mixed. You don't just want to put the specimen down into fixative and let it sit there. You want to gently mix it. Things that are more urgent, always our central nervous system, always. When we studied our central nervous system back in bacteriology, when we were talking about meningitis, we said that speed is of essence. Spinal fluids, anything from the central nervous system, is always a stat specimen. We want to process it quickly. We want to get it onto a slide for a gram stain. We want to, if it needs a wet mount for some reason, look at it that way. Get it plated, get it in the incubator. Get that gram stain stained in red if there's a gram stain involved. We never want to delay and batch that. We want to process everything quickly. And the parasites that do affect the central nervous system are very, very dangerous. We're going to learn they can kill you in a very short time. Another one that's stat is the blood films for malaria. We do diagnose malaria, babesiosis, leishmaniasis, usually leishmaniasis, but definitely plasmodium, which causes malaria, and babesiosis. Babesia, which causes babesiosis, we see those in blood films. We want to get those stained and red as quickly as we can. When we're talking about fecal specimens, there's different vials. You can have formalin, you can have polyvinyl alcohol, which is PVA. Usually the formalin's better for our immunological assays. The PVA is better for stain, but now there's generally one fixative called a total fix that is used. So formalin, we use formalin to look at the sedimentation of the eggs, the larvae, the oocyst. We can do a stain to look at cyst. We can do a trichome stain. There's a modified iron hematoxylin stain. There's a modified acid stain, other modified trichrome stains. When it comes to microscopic exams, it is sometimes hard to see the eggs. There may not be many there, even if we concentrate. So it takes careful and deliberate examination of that slide to be able to find these and then to identify the actual egg. So for our stools, you can look at the stool fresh, but usually it's preserved. Again, the live motile trophozoites can only be seen in fresh specimens. Once they're put in preservative or stained, then it kills the organism. We can still see it, but it cannot move at that point. It cannot exhibit the motility. There's cups that we'll look at in lab that have the formalin, polyvinyl, the alcohol. Barium, we cannot use barium. Some intestinal GI procedures or imaging services use a barium solution. You drink it, sort of a chalky substance, and it can obscure protozoa. If GI tests are done, colonoscopies, different things, and mineral oil is used, some antibiotics, if the patient is on antimalarial agents or antidiarrheal medications, they can sometimes obscure intestinal parasites. We're going to look at the stool macroscopically. We're going to see if it's liquid, if it's formed. We want to note that consistency. We want to look at it just kind of quickly to make sure we don't see worms or parts of worms. The liquid is best to see if there are trophozoites moving. Otherwise, usually soft or formed is where we're going to see organisms, not in a hard stool. We want to note in that macroscopic exam, is it bloody or is there mucus present? We do want to process these pretty quickly if they're non-preserved. 30 minutes for liquid specimens, an hour for semi-formed, 24 for formed. This testing has traditionally been called O and P. O for ova or eggs, and P for the other stages of the parasite. So the O and P exam. We're also going to get some duodenal aspirates. That's where they do the endoscopy, go down into our duodenum, the upper GI it's sometimes called. Maybe drainage, maybe mucus. There is an enterotest capsule where a capsule that is swallowed by the patient travels through the intestine. It's on a string, actually. Travels through the intestines on a string, and it comes out in the feces, and then it's checked for parasites. So with our O and P, we can do a direct wet mount, a concentration, a permanent stain. We can sometimes in the stool see some of the tapeworm body parts called the scolex. For pinworm examination, pinworm is caused by, the formal name is Enterobius vermicularis. With a pinworm, we do a tape prep. You can see the pinworm eggs in a stool specimen, but they deteriorate very quickly. But what happens is the female adult worm comes out of the anal opening at night usually and lays the eggs on the anal opening. So if we put a piece of tape or a sticky paddle on the anal area at night, the next morning take that tape or that sticky paddle, put it onto a microscope slide, look at it under the microscope, you can see the worms. We also have some molecular techniques and some immunoassays for our intestinal tract specimens. So with our fecal processing, we can do with formalin, we can do a trichrome stain, we can do immunological tests. They're called ELISA methods, and we're going to talk more about those. We can do a wet mount. With our PVA preservative, we can do trichrome stains. There's a formalin also that can be used, as formalin acetyl acetate sedimentation, that you can do wet mounts. You can do immunological fluorescence tests. You can do acid-fast stains and safranin stains. This is the pinworm exam. You take a piece of clear tape, put it on the patient's anal area, along the anal opening. The next day, generally, you take it off, you put the sticky side down on a clear glass, and you look at it under the microscope. Urogenital, we talked about Trichomonas vaginalis being sexually transmitted earlier, a venereal disease. So we can take vaginal or urethral discharge, prosthetic secretions, urine sediment, and do a wet mount procedure. We talked about that last semester, how with the wet mount procedure, if a person has genital itching, burning, discharge, we look for three things. We look for bacteria, Gardnerella, by seeing clue cells. We say clue cells are present or not. A clue cell is an skin epithelial cell obscured around the edges with bacteria. We are also looking for a parasite in those, Trichomonas vaginalis. It is motile. We see it moving in the specimen. And then a third thing we're looking for is a fungal infection caused by yeast. When we're looking for the Trichomonas, so three different things. Let me go back in that wet mount, that saline wet mount procedure. The bacteria Gardnerella, clue cells. The yeast, we see the actual yeast forms, and we'll talk about that with our mycology. And the third thing is this parasite, Trichomonas. They have to be motile. We want to look under low power, maybe go up to high. We never remember with the wet mount go up to the oil immersion lens, only low and high. We want to turn our light down, and we're looking for motile organisms. If we do not see a motile organism, we do not see it moving, spinning, swimming, then we cannot call it, even if we see something that looks like it, if it is dead or not moving. Also, there's a very good direct antigen test for Trichomonas that can be used, but this is a really quick way, and we'll talk about it a little bit more when we get to the organism. Sputum. They're not always found in the sputum. It's rare, but there are a few we're going to talk about. Paragonimus, a few of the others listed here that do go through the heart-lung cycle. We can sometimes see those in the sputum. They can cause pneumonia, other things. We won't have an induced sputum, so make sure it is a good coughed up sputum to see these. Liquid specimens. We get liquid specimens from fine needle aspirates, from that endoscopy duodenal aspirate, from bronchial lavages, washings, if we collect bone marrow, if we collect spinal fluid. We can get fine needle aspirates of cysts, of abscesses. We can stain those to look for amoeba. There's different stains for those. There's a Giemsa stain, a modified trichrome stain. And we can stain bone marrow aspirates to look for different organisms. We can take those bronchial specimens. Those are listed here, duodenal aspirates, spinal fluid. There's several stains that can be used, and we'll talk about some of these different organisms as we go through those. Biopsy. Sometimes we have to get a tissue sample. If there's a parasitic infection in the tissue, we can make smears, do what's called a tease mount, where you pull that tissue apart and even kind of press it between two different surfaces to kind of squash it, to look at that under the microscope. With our blood, you could see the parasites in just whole blood with a cover slip, but generally we make a stain. These are stains just like your hematology stains are prepared, and if the doctor suspects one of these protozoa that can be found in the blood, generally based on the patient's travel history, they're going to request this. We make a thick and a thin smear from whole, fresh blood, either whole, it's always whole blood, I'm sorry, it can be fresh, or it can be from an EDTA tube. The best way is from a finger stick. We'll talk about with the malaria how when their fever spikes is when it's circulating in their bloodstream more. So when the fever spikes, the nursing unit should call the lab to come up, do a finger stick, drop it onto a slide at bedside, put a drop on several slides, and just let it kind of pool out on its own. That's our thick smear. and then put a drop near the end and make some thin smears at the bedside. The alternative is to draw that EDTA tube and make the smears immediately when it gets back to the lab. The thick shows the quantity because there may not be many there, so the thicker it is, but you can't always see the forms or the shape. So that's just to see something and be a screen. Then if you see something, you go to your thin smear to see the features. The CDC recommends we make at least 2 sets, but you're probably going to make more just to be on the safe side. Also, we'll go back up if the patient's blood spikes again, blood spikes. If their fever spikes again, maybe go up every so many hours and collect more specimens. So on the thin blood, the thick, we're going to start with the thick. I don't know why they did the slide this way. We're going to look at the thick first under low power, then go up to oil to look a little bit closer. Then if we see something, we're going to go to our thin, look at low power, then go to our, usually go up to our oil and look also, but you can see most under low power. Just depends. You want to examine at least 300 fields before reporting it negative. You want to look at your feathered edge along with your thick areas, just like you do with your hematology counts. And when you look at those, with your hematology counts, you're a lot of times counting white blood cells in a little bit thicker area, but you want to look at your red cell morphology in thinner areas. So kind of like that, you want to look everywhere. 300 fields can be hard to count, so one rule of thumb is kind of like with our mycobacteria, if you remember, instead of trying to count 300 fields, spend at least 15 to 20 minutes examining the slide. And that's usually about the same thing. Okay, so we start with the thick. I got ahead of myself. You look for adequate portion of white blood cells. If you tentatively see something, maybe get a species idea, then you go to your thin smear. If it's suspected, but you don't see it in the thick smears, if you suspect malaria, but you don't see anything in the thick smear, you cannot say no parasite seen or you've done those 300 fields with at least 20 white blood cells or that 15 to 20 minutes. Again, the CDC recommends, and most hospitals have a policy that you don't stain but one set of your slides. Then when you send it to the reference lab for confirmation, you usually send some stained slides and some unstained because they a lot of times like to stain their own. We can use our right stain like we use in hematology, our Giemsa stain. You can do a Buffy coat. You can take the white blood cell layer of your peripheral blood and just make a smear of that instead of making a smear of the whole blood. Leishmania donovani, our trypanosomes, and our histoplasma capsulatum, which is a fungus we're going to talk about later that's seen in the blood, are usually seen more in the white blood cells. With our intestinal parasites, we can use that antigen detection. This is something, it says are not used routinely in the clinical lab because of sensitivity, specificity, and expense. But there have been, and this is talking about direct fluorescent antibodies, enzyme immunoassays, immunofluorescent, but there have been some that have actually been developed in the last few years that are rapid and very easy to interpret that will identify our Giardia and our Cryptosporidium. Giardia and Cryptosporidium are two of our main parasitic infections we have seen in the intestines in the U.S. We do see some entamoeba, but not a lot. But it's very easy to screen for Giardia and Cryptosporidium using one of these rapid direct detection assays. And a lot of hospitals do these. With our blood parasites, there are some antigen methods to detect these. There are some PCR kits that will detect Plasmodium that causes malaria. There's some molecular techniques. Most of these, even though we're starting to see more molecular testing done in the clinical laboratory, these types of tests you're generally not going to see in the clinical lab. There'll be research in reference lab. This was mostly developed for trypanosomiasis, amoebiasis, cryptosporidiosis, giardiasis, leishmaniasis, and microsporidial infections, malaria, and toxoplasmosis. Polymerase chain reaction PCR are used at the CDC to detect the PCR procedures, detect Giardia, Cryptosporidium, Cyclospora, Entamoeba, histolytica and dispar. With the ova and parasite, we already talked about that just a little bit. We do the direct wet mount on fresh. The concentration, you do an indirect wet mount after you've concentrated it. A lot of times this has replaced the direct wet mount. We don't do the fresh anymore, so we concentrate it, then we look at it, and then after we've looked at it concentrated, we look at the sediment generally and the supernatant, the top part. And then we take it and we do a stain on it after we've looked at it wet. There are instances where you can culture feces for larvae. Sorry, I had to pause for just a minute. Culture, sometimes after concentration you can't detect an organism. It does, if it's cultured, allows the larvae to develop into other stages called falciform stages, but it's very, very complicated. It requires a lot of quality control, a lot of very specific steps, very, very time consuming. So it's generally used for research, not for any kind of diagnosis, but to research the development of the organism, to see changes in the organism. There's one organism called acanthamoeba that can be actually cultured onto a nutrient type agar, but then you have to overlay it with E. coli or Enterobacter to be a food source. Then you have to do PCR testing after that. So it's all very complicated and not what we consider a normal culture process like we did in bacteriology. There are some Trichomonas vaginalis test kits for a quick culture type method, but again, these are rarely ever used. We do use Trichomonas antigen testing kits, but not usually for culture confirmation. Most of our protozoan parasites are not cultured at all in laboratories. For serodiagnosis, this is drawing the patient's blood and looking at testing their serum. Again, these are very expensive. We usually don't do these because they are so expensive. They're hard to interpretate. They have a very low sensitivity and specificity. But there are some reference and research labs that do serodiagnosis. With toxoplasma, sometimes it's recommended in case the infection is very widespread and you're trying to look at epidemiology and see where that has spread. So sometimes that can be used by the CDC. There's latex agglutination testing that does these, the ELISA, the EIA, the enzyme, the immunofluorescent assays, and the immunoblot techniques. Prevention with parasitic infections. How are we going to prevent these? A lot of times it is personal hygiene and sanitation. Eliminating sexual activities that potentiate fecal oral contact. With sanitation. In the United States, we have very good water filtration. We have sewage filtration. So generally, these things are not in our environment. If we are out in the environment, we want to just be very careful with water. We think when we go out hiking, camping, that spring water, stream water is natural, it's clean, it looks clean, it's bubbling along down through a mountainside or wherever. But there can be animals upstream that have their fecal contamination has gotten to that water with parasites. They make a tablet when you go hiking, camping that will sanitize the water, but it will not get rid of two organisms we're going to study. Giardia and Cryptosporidium. They will not be killed by those tablets. They have to be filtered out. In fact, they would be in our city water supply if we just treated it with chemicals. Our city water supply is filtered to filter those out, and it is checked to make sure those are being filtered out. Another way to get some of those is well water, things like that if it's not treated. So that's some of the preventative measures that we can use. being very careful with, we're going to study some organisms that get into our contact lens cases. Being careful out working in the soil with sanitation. Um, Malaria. How do we prevent malaria when we go to places where it's endemic, like Africa? There are screens that you can put up. There are ways to keep from being bitten by mosquitoes to control some of those vectors. Um, we'll start with the chemoprophylactic. I'm sorry, there's medications you can take, but also if you think you're exposed to it, you can take some prophylactic type antibiotics. Vector control. Again, when you travel, you can use screens and ways to prevent from being bitten. Wear protective clothing. Thinking about other bites out in the environment. Awareness of blood transmission. Several of these are transmitted through blood, so we want to be careful with blood transfusions. We're going to talk about some of these that are checked for in our blood supply. We'll talk about that now. We'll talk about it next fall in our blood bank class. Not sharing drug needles. Some of these we have to make sure with organ transplants that the organs are checked, the donor is checked. Some of these are congenital, can go mother to baby. Other methods are meat. We're going to learn about some parasites that can be avoided by cooking our meat, just like we talked about in bacteriology. Some of our enteral hemorrhagic E. coli could be prevented by adequately cooking the meat. Same thing here. There's parasites that as long as we cook the meat correctly, handle the food well, do not touch our mouth with hands that have touched raw food, things like that, then they will not be transmitted. With our so that's kind of transmission. There is one worm we're going to talk about briefly, the guinea worm, that the World Health Organization actually and the CDC actually partnered to clean up drinking water worldwide, to get rid of an organism in the water called the guinea worm, Dracunculus medinensis. And we'll talk about that, but it's basically been eradicated because of their campaign to clean it from the drinking water worldwide. Okay, so when we start to look at our ectoparasites, the arthropods, the ectoparasites are part of the phylum Arthropoda. They're biological vectors of a lot of disease. Mosquitoes transmit malaria. Ticks transmitted Lyme disease. We talked about Lyme disease back with our bacteria. Remember, that's a bacterial infection. So biological is when they actually have the organism in their body and they transmit it to us. But also we discussed mites, lice can cause disease, but also the mechanical vectors I mentioned earlier. The fly, the cockroach lands on your food or your water. We're going to talk about some that land on your skin and can just break your skin, and it may not actually be living in that arthropod, but it's on their wings, it's on their legs, it's somewhere on their body. So again, we're not necessarily going to study these actual ectoparasites, but sometimes something may be brought to the lab. We've had body lice, we've had scabies, things brought to the lab, and we've just put it under the stereoscope just because the infectious disease doctor wanted to look at it, but generally we're not identifying that as such as part of the diagnosis. They just may want to know, yes, is that what they're seeing on the patient's body or not, so they can kind of control that. We did find one time in one of our wet mounts here in the lab, we found an organism that looked like an arthropod that was in the stool specimen. Of whether it came from the patient from the stool or got into the vial, we don't know. But some of those pictures are in your book. A Rocky Mountain wood tick that caused a disease, a bacterial disease that we talked about earlier, the rickettsia. We've just covered that. There's a rat flea that causes typhus. We talked about that already. Then there's a louse that causes different diseases. So again, they don't just cause our bacterial infections. We're going to talk about some parasites that they cause. Again, we're not going to look at this so much. This is the makeup of the arthropods. It listed in your book. They have an exoskeleton. They have jointed legs or appendages. Some have more than one pair. Some have 4 appendages. Sometimes have 3 pair. So it's going to be different numbers for that. Talk about the bilateral symmetry of them. But again, we're not going to really study those in the clinical laboratory. This does list the types: the arachnida, ticks, mites, spiders, scorpions; the insecta, flies, lice, fleas, bugs, and mosquitoes. So that ends our introductory chapter. Next, we'll move into our intestinal protozoa, then our, I think next after that is our blood and tissue protozoa, and then the last chapter is protozoa from other body sites. So we'll cover those in the next couple of lectures. So this is that link, that CDC website, DPDX. Just one example. The main thing we're going to use is this A through Z index. And to start out with, just quickly, one I'll show you. Malaria. Malaria is caused by an organism called Plasmodium. So I can click on M for malaria, and I can pull it up here, or I can go down here and I see the different Plasmodium species: Plasmodium falciparum, malariae, ovale vivax. There's 5 of those here. We can click on any one of those, or just the malaria. It's going to show all of that to me. When you get in here, you start out with the parasite biology. We see the causative agent. We see the life cycle. We're going to go through these when we go through the organisms. It explains that whole life cycle. It gives us a geographic distribution, a clinical presentation. Then when we go up to image gallery, this is very important. We're going to use these images as we study the organisms. We're going to see the rings. This is in a thick smear. Looks like a little ring here with a little dot. And then we also see this is a thin smear. You can see that inside the red blood cells. You can see that ring form inside the red blood cells. And then we can also see the gametes. This is showing the actual little gamete forms in the blood. As part of the reproductive cycle, the gametocytes, not gametes, I'm sorry, the gametocytes. So different objects that we can see in our blood smear. There's also a laboratory diagnosis section and a treatment. We're not going to the treatment information we're not going to dwell on very much. We might mention it in a few cases, but we are going to talk about the image gallery quite a bit, the life cycle, and some of the lab diagnosis. So very good website. Then you just go back to your A to Z index, pick another organism. So very, very good website. A lot of good reference for you as we study this. Also, there's pictures in your book. Again, the pictures in this chapter, they show quite a few pictures, but we are going to see those pictures when we get to those organisms. We may refer back to those because some of those I think are really better in that chapter. We'll kind of go back and forth on those as we study this. And near the end of the semester, it's on your schedule. It's sometime in April. We will have what's called a wet mount practical that will include parasites and some of our fungi, some of our yeast. And we will have some pictures, a picture slideshow for part of that, and we'll have some wet mounts under the microscope. And we'll be talking about more of those in lab. So next will be our intestinal protozoa.